से पहले अगर पार्टिकल फिजिक्स में किसी को प्रॉब्लम है ग्रेविटेशनल वेव्स या ब्लैक होल्स में तो मुझे बताओ। कोई भी प्रॉब्लम, कैसी भी प्रॉब्लम। Yes, Subhajit, bosons, they have mass. They can have mass. Bosons are not the forces. What we are saying in particle physics, we are saying that there are 4 fundamental forces in nature, and all the particles in the universe, okay, they interact by one of these interactions. Okay, in case of electromagnetic interaction, we have one electromagnetic field. There is electromagnetic field and 2 particles interact in this field by exchanging photons. The mediator is photon. Okay, and this photon, it is also a boson because its spin is integer spin. Photon is a boson. And photon is massless, but in case of Higgs boson, when the particles they interacted with the Higgs field, okay, so when the particles they interacted with the Higgs field, then WZ bosons, the Higgs boson was the mediator particle, and Higgs boson, it has mass of approximately 126 GeV, giga electron volt. गुड इवनिंग विपुल, काफी लंबी लड़ाई थी। ओके, तो पार्टिकल फिजिक्स में फंडामेंटल फोर्सेस को एक्सप्लेन कर रहे हैं और इंटरेक्शन, आई एम गुड विपुल, and the particles are interacting by exchanging particles. Okay, in case of electromagnetic interaction, the exchange particle is photon. In case of weak interaction, we have WZ bosons. Okay, in case of strong force, we have gluon. And standard model, it does not cover the gravity, and for the gravity we have proposed graviton. Okay, and Higgs boson is because of the Higgs field. Okay, the particles they interact with the Higgs field. When the universe expanded after the Big Bang, all the particles were massless and they interacted with the Higgs field, and because of that interaction they acquired mass. Okay, so they are interacting with the Higgs field via the Higgs boson. Formation of mirage by TIR. Yes, I think in case of mirage also, yes, there is TIR. See, in case of weak interaction, okay, in case of weak interaction, the W and Z bosons they will be involved, and in the weak interaction, the neutrinos are produced. Whenever in any reaction, okay, the neutrinos they are produced, then that is the weak interaction. Okay, Arpit? Whenever any 2 particles are interacting through weak interaction, they are exchanging the W and Z bosons. Okay, and it is a fact that neutrinos they are. Happy birthday, Arya.
Thank you, sir. How do you know?
The management knows everything.
Okay. Thank you, sir.
Okay, enjoy your day. Okay, so neutrinos, they are produced in the weak interaction. In beta decay is the classic example of weak interaction. Beta decay cannot be explained by the electromagnetic interaction or any other interaction. To explain the beta decay, we have the neutrinos are produced in the beta decay, and W and Z bosons, they are the mediator particles in the weak interaction. And the 2013 Nobel Prize, the Nobel Prize of 2013, it was given to Peter Higgs. Okay, Peter Higgs was awarded this Nobel Prize of 2013 Physics for the discovery of Higgs boson. So 2013 Nobel given to Peter Higgs, and there was also one more fellow with Peter Higgs, and you have to verify his name from Google. I don't remember his name, but Peter Higgs was awarded Nobel Prize for the discovery of Higgs boson. And this Peter Higgs is the person who proposed the Higgs field, who proposed that theory that in the starting of the universe, after the Big Bang, all the particles were massless and they acquired mass by interacting with the Higgs field. This theory was proposed by Peter Higgs, and Peter Higgs also proposed that the mediator particle in this case is a boson, and the mass of that boson, it should be around 126 GeV. And in the Large Hadron Collider in the CERN, Geneva, okay, a particle was discovered and whose mass was 125 GeV. That confirmed the existence of Higgs boson, and that validated the theory of Peter Higgs. Okay, and Peter Higgs inferred this from the standard model of particle physics that there should be a Higgs field. Now we are starting with the biotechnology.
Sir, just a question, sir.
Sorry, Arpit?
Sir, a question, sir. A query, sir.
Yes, please.
Sir, it means, sir, during the alpha and beta decay, so both this neutrino, and WZ boson, both will be produced, sir.
The W, they are interacting by the exchanging of W and Z bosons. Okay, and we only see in the reaction the product is the neutrino. Okay, W and Z bosons, they will be produced and they will vanish. The interacting particles, okay, they are mediating through the W and Z bosons, and when the interaction is over, the W and Z bosons, okay, they will also vanish. So what the scientists they do at the Large Hadron Collider, they are continuously colliding the particles with high energy with each other, and in the interaction these particles are produced for a fraction of a second. Okay, and they are measuring the masses of these particles.
Right, sir. Sir, what are these intermediary particles? Sir, I didn't get it, sir.
Arpit, see the quarks. Okay, quarks are fundamental particles.
Yes.
Okay, so the quarks, they are they bind together by the strong force. Okay, the quarks, they are held together by the strong force, and the strong force arises when they exchange the gluons, the particles known as gluons. This is the proposed theory that two particles are interacting by exchanging these mediator particles, gluons, and because of this interaction the force is arising.
Okay, sir.
अभी तक हम कैसे करते थे, न्यूटन ने कहा था that gravity, okay, there is gravity and gravity attracts things. यहाँ पे हम कह रहे हैं कि because there is a force, that means two particles they are interacting by exchanging some particles, and those particles In case of strong force, they are the gluons. Okay. In case of electromagnetic force, there are photons. In case of gravity, they have proposed gravitons. And in case of weak interaction, we have W and Z bosons. This is the proposal of the theory. ये थ्योरी ने प्रपोज कर दिया, ठीक है, थ्योरी से ये चीजें निकल के आई और ये सब चीजें टेस्ट हुई हैं CERN में, जो लार्ज हेड्रोन कोलाइडर है। और जब हम बोmbardment करते हैं पार्टिकल्स का, प्रोटॉन एंड प्रोटॉन दे कोलाइड, देन इन दैट प्रोसेस these particles are produced, okay, and scientists have they have identified these particles like the gluons, the WZ bosons, and finally they also discovered the Higgs boson. So these particles, they exist.
Okay, sir, got it. Sir, only one last query, sir. Sir, this
Yes, sir.
fraction spin at integer
Yes, the spin.
Yes, sir. Spin,
Yes.
sir, this fraction spin is rated to fermions and this integer spin is rated to bosons.
No, we have classified those particles which are having the fractional spin. Those particles, they are termed as fermions. And those particles which have integer spin, they are termed as bosons. So this is the classification on the basis of this property which we call as spin. Okay, the classification in fermions and boson, it is on the basis of spin.
Okay, sir, half spin means half का spin का क्या मतलब है? मतलब आधा ही घूम रहा है सर, मतलब क्या?
No, वो rotate ही नहीं कर रहा है. This is altogether quantum mechanical concept. Okay, and I would suggest you that separately तुम इसका पूछना, इससे class extend ही होगी.
Right.
See, in the classical world, the world that we witness in our everyday life, we consider the spinning as something spinning, rotating. Okay, but in the quantum mechanical world, this is not related to rotation. It is a separate property, but it is related to the angular momentum. The dimensions are same as that of the spinning, So don't go into it. It is related to the quantum mechanics. Now Nitin is asking, sir, can we say that space has curvature due to this property of fundamental particle? No, the curvature of space, it is related to the mass. Okay, in the Einstein theory, he has proposed that energy and momentum, mass is a form of energy, energy and momentum, they are responsible for curvature of space. Okay. Now we are starting with new topic, which is the biotechnology. And yes, the second person, the one person was the Peter Higgs, and the second person, along with Peter Higgs, was awarded the Nobel Prize of 2012. He was Francois Engler. Okay. Now, what is this biotechnology? Okay, biotechnology is that branch of biology that utilizes the biological systems. Okay, the branch of biology that utilizes the biological systems or the living organisms, okay, or any of the part of the living organisms to develop or create new products. Okay, so this is biotechnology. It is the branch of biology, and in this, okay, in this technology, we are using the living organisms, we are using any biological system, and even we are using the parts of the living organism to produce some products. So this is biotechnology, and the crudest biotechnology we all encounter in our everyday life. Okay, dahi sabne jamai hai ghar pe, the curdling of milk. The curdling of milk using the bacteria, that is also some sort of biotechnology. Okay, the crudest form. Okay, the producing of alcohol by fermentation of sugar, that is also a sort of biotechnology. But now today we have entered into this sophisticated world of biotechnology. Okay, Lactobacillus. Yes, people, very good. Vipul, mass, energy, and momentum is responsible for curvature of the space. Okay, mass, energy, and momentum, they modify the geometry of the space, and that manifestation of the space is gravity. Gravity is not okay something separate. Gravity, we are calling the curvature of the space as gravity. Gravity is nothing but, gravity is not a force. The curvature of the space is gravity. न्यूटन कहता था there exists some force, but Einstein ने कहा no, the space is curved. Okay. Is it fine, people? Okay, very well. Now the baking of bread and the brewing, okay, all of this is biotechnology. And baking of the bread, it obviously uses the yeast. So biotechnology is that branch of biology where the new things are developed using living organisms. So we are starting with the DNA. Okay, we're starting with the DNA, and the most common, the building block of, okay, the building block of the any living organism, they are the cells. Okay, the building block of macroscopic organisms, they are cells. Aishwarya is typing a question, so let's wait for her question. Yes, Aishwarya, very good. The DNA is deoxyribonucleic acid. Very well. And I am talking about the building block of the organism. They are the cell. Okay, cell has the crudest cell. This is the diagram. The सबसे जो मोटी-मोटी बातें हैं. The outer membrane is the, this is the cell membrane, and it confines all the cellular material in a given volume. Okay, so we have a cell membrane, and through the cell membrane, the cell can interact with the surroundings by some processes. And inside a cell, we also have a nucleus. Okay, you can crudely call it at the brain of the cell, and we have some fluid in the cellular space, and that fluid is known as cytoplasm. Okay, this is the most simple schematic diagram of a cell. Okay, and this is a more detailed diagram of a cell, and we do not need to go into the details. Okay, so there are certain chemicals. This cell is made up of organic molecules. Okay, this cell is also made up of the fundamental particles that we have discussed in the last class. Vipul, the mitochondria is the powerhouse of the cell. Mitochondria is the powerhouse of the cell. Okay. Good evening, Nikhil.
Sorry, sir. I clicked on wrong link, sir. So that's why I couldn't join.
It's okay, Nikhil. Don't worry. I was telling the guys about the biotechnology. Okay, so today we are starting with biotechnology, and I have just explained that biotechnology is the branch of biology where the new products are developed. Okay, using biological systems like the living organism and the parts of living organisms. Okay, Nikhil. So this is the scope of biotechnology. In biotechnology, we are producing new products using living organisms. Is it clear to you?
Yes, sir. Thank you, sir.
And the most common example is the curdling of milk. Because when the milk, it turns into a new product, which is curd. And in this process, okay, certain bacteria, they are used to convert the milk into curd. So this is the crudest form of biotechnology. Okay, and now we will discuss some sophisticated biotechnology. Okay, and I have explained the class that the building block of organism, they are the cells, and cell has a very common structure. Cell has a cell membrane, which is basically confining the cellular material inside a given volume, and the cell also has a center, which is the nucleus of the cell, which acts like a brain of the cell, and the space inside the cell is filled with a fluid which is known as cytoplasm.
Thank you, sir.
Okay, so welcome. So there are certain chemicals which are present inside the cell, and these chemicals, they have the basic properties. Okay. We have acid and bases, so we have these base molecules inside the cell. And there is a category of organic molecules. There is a category of organic molecules which are known as nitrogenous bases. Please write everything as I am speaking, write everything that is present on this slide, and you can obviously skip this diagram. This is not of your use, but you have to write all these things. The nitrogenous bases, these are important. We have adenine. Okay, these are basically organic chemical compound containing nitrogen. Okay, and they have the basic properties. So we call them nitrogenous bases. And these nitrogenous bases, they are of various types. We have the A is adenine. We have T is thymine. We have guanine, G for guanine, and we have cytosine, and we have the uracil. So these are the nitrogenous bases present inside the cell, and these nitrogenous bases, they are simply the chemical compounds. Okay. Now please remember some things, like the nitrogenous base plus sugar, this is known as nucleoside. Okay, and if you will add one more thing, that is the phosphate. The nitrogenous base plus sugar plus phosphate, this is a new thing which is known as nucleotide. Okay, nitrogenous base plus sugar, this is nucleoside. Nitrogenous base plus sugar plus phosphate, that is nucleotide. Okay, and a chain of nucleotide, when several nucleotides they are connected, a chain of nucleotide, that is known as nucleic acid. So we ने ये term सुनी होगी, nucleic acid. Okay, Ruxana, have you received your copy? The checked copy?
Yes, sir, I have received it.
Okay, and Meenu, have you received your copy? Okay, you haven't. Okay, I will check it. Okay, means I have already evaluated it, but I will ask to the management what has happened. I evaluated it the day before yesterday, or I think I evaluated it yesterday. So the nucleic acid, it is the chain of nucleotide. Okay, ठीक है. अब सबको टर्म्स क्लियर है. नाइट्रोजनस बेस, न्यूक्लियोसाइड, न्यूक्लियोटाइड एंड न्यूक्लिक एसिड. Is it clear to everyone? Very good. Now we have two kinds of nucleic acid. There are two types of nucleic acid. One is the DNA and one is the RNA. Okay, and अभी एक बच्चे ने बताया था DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Now there is some difference between these two kinds of nucleic acid. Okay. Yes, Ashwarya, very good. In case of DNA, the sugar that is present in the DNA molecule, it is the deoxyribose sugar, and the sugar that is contained in the RNA, it is the ribose sugar. This is one difference. Okay. The second difference is in DNA we have the adenine, we have thymine, we have guanine, and we have cytosine. But in case of RNA, we do not have the thymine. Okay, and instead of thymine, we have the uracil, as Aishwarya has pointed out. Okay, the difference is of the thymine is replaced by uracil. And the last difference is DNA is double-stranded and RNA is single-stranded. So these are two strands of DNA and this is the single strand of RNA. Okay, and here they have shown the chemical compounds. This is the chemical structure of thymine. You do not have to remember it. And this is the chemical structure of cytosine. Okay. यहाँ तक सबको clear? What is the difference between RNA and DNA? Nitin is giving extra information. He is saying that purines A plus G is purines and pyrimidines is G plus C. Very good. Now there is one more thing. In the double strand of this DNA, okay, the one strand is connected to another by way of some bonds, the hydrogen bonds, and the A it will always pair with T and G will always pair with C. The adenine it will always pair with thymine and guanine it will always pair with cytosine. Okay. So if we have a chain like this, we have one strand where we like if we have the chemicals arranged in a strand like this, okay, then in the other strand it will be linked to A will be linked to T, T has to link with A, C has to link with G. Okay, so these are two strands. This is strand 1 and this is strand 2. Okay, T has to bind with A, G has to bind with C, A has to bind with T. So this is strand 1 and this is the strand 2. A always combines with T and G always combines with C. Yes, Minu. They are joined by the hydrogen bonds. Okay, Palak is saying that we have two hydrogen bonds between A and T. You can also write it. There are two hydrogen bonds between A and C, and there are three hydrogen bonds between G and C. Okay, but A will always pair with T and G will always pair with C. That is the thing. So the strand of the DNA, the strand is made up of sugar and phosphate. This base, okay, it is made up of sugar and phosphate, and these links, these strains, they are basically adenine is joined to thymine, guanine is joined to cytosine by the usage of hydrogen bonds. So this is the double helical structure of DNA. And each cell contains the 6 feet long DNA. And I have already told you that A will pair with T and G will pair with C. Okay, so now write one factual information that who gave the helical structure of this DNA. So we have James Watson. Yes, Watson and Crick. We have James Watson and Francis Crick. Okay.
So can you repeat the name again?
It is James Watson and Francis Crick. So DNA is double-stranded and RNA is single-stranded. Yes, sir. Now let us move on to these chromosomes. See we have the DNA. This is the DNA strand, double strand of the DNA. Okay. If you will compress it, इसको compress कर देंगे condensed form में, then it will appear something like this. Please draw this diagram, and the condensed form of DNA, this is known as chromatin. So this is chromatin. Okay, but this chromatin, it is still very long. After condensing it to a smaller form, it is still very long. So if you will compress it to the maximum strength, okay, if you compress it to the maximum amount, then it will appear something like this. Nikhil, I have drawn a very long strand. Okay, consider like
Okay.
we have, okay, a very long strand. We have two threads which are very long, and the threads, they are connected. Okay, and you have wrapped these threads. Okay, and you have shown by wrapping them, you have shrink them to a smaller volume. So DNA is like a long thread, and you have converted it, you have transformed it into a smaller volume. Okay, and that is known as the chromatin.
Thank you, sir.
You have collected the threads together and you have compressed them, so that collected form is known as chromatin. And when you, okay, the maximum condensed form, that is known as chromatin. The maximum condensed form is chromatin. Okay, Aishwarya is saying we have euchromatic and we have the heterochromatic. I think we have 2 doctors in the class. So what you are asking the use of this? See, we are not doing this. This is the natural process that is happening inside our cell. We are not creating these inside the labs. Okay. We have observed these things inside the labs. Chromatid is the maximum condensed form. Okay, DNA के thread को आपने the maximum condensed form of the DNA. If you compress the chromatin further, then we have this form which is known as chromatid. Simply put, DNA को उठा के लपेट दिया आपने, उसको छोटा कर दिया और उसको जब मैक्सिमम जितना छोटा कर सकते थे उतना कर दिया. That is the chromatid. Very well. उठा के लपेट दिया DNA को, उठा के लपेटा, we have the chromatin and उसको थोड़ा properly arrange करके minimum volume के अंदर लपेट दिया. That is the chromatid. And the two chromatids, they join together to form the chromosome. The two chromatids, they combine to form the chromosome. So this is a chromosome. डोंट वरी, बायो से मेरे ख्याल से पिछले 6-7 साल में कोई क्वेश्चन नहीं आया. नो क्वेश्चन वाज आस्क्ड फ्रॉम द बायोलॉजी इन द लास्ट 6-7 years. So the questions they will be asked related to the biotechnology. Yes, Kritika, that DNA, the 6 feet long DNA, it is compressed to the form of chromatid. Okay, so each of the cell, the building block of organism, each of the cell, it contains the 23 pairs of chromosomes. So one chromosome is a combination of 2 chromatids. This is one chromosome, and there are 23 such pairs. So in total, we have 46 chromosomes inside the cell of the human body. Okay, so Palak is saying that the maximally compressed form is the chromosome, and the 2 strands of the chromosome, they are known as chromatids. Just remember this diagram, you will compress the DNA, and from the DNA, the chromatin is produced, and finally, we have the chromosome. Okay. It is the same thing. Vipul, we are just condensing. एक रस्सी को लपेट दोगे, okay, that is the same thing. उसको खुला कर दिया and उसको लपेट के छोटा कर दिया. Okay, जब उसको खोल रखा है, that is the DNA, लपेट के छोटा कर दिया, that is the chromatid or the chromosome. इतनी सी बात है. पुराने टाइम में जो ऑडियो टेप्स होती थी, ऑडियो टेप्स को कभी खोला है? उसके अंदर से जो रील है, मैग्नेटिक टेप, ओके, दैट इज इन द कंडेंस फॉर्म, छोटी सी वॉल्यूम में उसको श्रिंक करके रखा हुआ है, एंड उसको खोल दोगे तो कितना ज्यादा फैल जाएगा। सो सेम थिंग यहाँ पे है। So the condensed form of DNA is chromatin, and chromatin is, okay, it is still a very long molecule. DNA is itself 6 feet long, and chromatin is still a very long molecule, and this chromatin is further compressed, it is condensed to form the chromatin. Yes, Nikhil, the audio cassettes. The maximum condensed form of DNA is chromatid, and two chromatids, they join to form the chromosomes. Okay, so this is the structure of the chromosome. Okay, and there are 23 pairs of chromosome in each cell of the human body. 23 pairs, so we have total of 46 chromosomes. Very well. Each cell contains 23 pairs of chromosomes, and 22 pairs of these 23 pairs, they are known as autosomes. The 23 pairs, that is the 44 chromosomes, they belong to the category of autosomes. AUTOSOMES. Okay. Reed, you wanted to ask something? Yes, 23 pairs. 23 pairs, so 46 chromosomes. The 22 pairs, that is the 44 chromosomes, they belong to the category of autosomes. And the 23rd pair, okay, 2 chromosomes, they are given a separate name, which is the allosome. And the allosomes, they differ from autosomes in shape, size, and behavior. Okay, and this 23rd pair, the last 2 chromosomes, they are known as the sex chromosomes or the allosome. Okay, and these chromosomes, they are responsible for determining the gender in an individual. Okay, the newly formed individual will be a male or a female. It depends on these 2 chromosomes. Yes, number of chromosomes, okay, they may vary. Okay, so the allosomes or the sex chromosomes, they determine the sex in an individual. Okay, the gender of an individual is determined by these chromosomes, and they are given a separate name, which is allosomes. Okay, so we have autosomes and we have the allosomes, and allosomes, they serve a specific purpose. They determine the gender of an individual. Okay, so these 22 pairs, the autosomes, they are classified on the basis of their size. The largest one is named as the first autosome. Okay, then the second largest is the second autosome, and likewise we have arranged them in descending order, in the descending order of their size. Okay, so the 22nd pair of chromosome, the 22nd autosome, it is the smallest one, and the first autosome, it is the largest one in the size. Okay, So these allosomes, these sex chromosomes, they are of two types. ये तो सभी ने सुना होगा, the X and Y. Okay, the sex chromosomes, they are of two types, X chromosome and Y chromosome. So if the 23rd pair, it is X and X. Okay, the chromosome number 45 and 46, if it is the X and X, then the individual will be a female. And if the 23rd pair is X and Y, then the individual will be a male. Okay. Now in the females and in the birth process, see the females, they are producing eggs, and eggs they are a special type of cells. Okay, the eggs and sperms, they are a special type of cells and they contain only 23 chromosomes. Okay, इनके अंदर 46 chromosomes ना होके सिर्फ 23 chromosomes होते हैं, and we have these special cells which are egg and sperm. So egg is produced inside the female body and sperm is produced inside the male body. Okay, and the egg, it contains 23 chromosomes, and sperm, it also contains the 23 chromosomes. So half the number of chromosomes, they are present in egg and sperm. Okay. Now what will happen when the sperm, it fuses with egg? Okay, so suppose the egg, it contains the 22 chromosomes, and in females it will have the X chromosome because females have only X and X. So इस पेयर में से एक X इस egg cell के अंदर जाएगा. Okay, out of the 46 chromosome, 23 egg cell के अंदर होंगे. So 22 will be the autosomes and one is X, the allosome. Similarly, in the sperm cell, we will have 22 autosomes and there will be one. Okay, there will be one chromosome. It will be either X or it will be either Y. So the sperm, it may contain either 22 plus Y or it will be either 22 plus X. Okay. And female, they produce only one kind of egg cell, which is 22 plus X. Now, if the sperm cell containing 22 plus Y, it will fuse with the egg, then the zygote that will be formed is the fusion product. It will contain 22 autosomes from the female, 22 autosomes from the male, and it will contain the X and it will contain the Y. And because it contains X and Y both, it will be a male child. Okay, so this is product of this one and this one. But if 22X Little Palak can tell you, okay? Palak is a doctor and she can tell you that two microorganisms also have the allosomes. Yes, in microorganism, the fertilization process is different. Okay. Basically, they replicate by asexual process. See, in answering these kind of questions, new questions will arise. Here the term like the fertilization term appears, the gametes. Okay, these terms appear. So Palak has to use these terminology to explain you something. Okay, so please stick to what I am teaching you in the class. Okay, and if a question is related to it, then please ask it. So if the sperm containing 22 plus eggs, it will fuse with the female egg, then in that case we will have a female child. Okay, so 22 autosomes from the male, 22 autosomes from the female, and we will have X from the female and X from the male, and this will be a female child. Okay. So the sperm containing 23 chromosomes from the male, it fuses with the egg of the female containing 23 chromosomes, and these egg and sperm, they are different kind of cells. Okay, and they contain only 23 chromosomes. Okay, so the fusion of egg and sperm, it produces the zygote. So the fusion product containing 22 plus 22 and X and Y. Okay, so this product is known as zygote, which is formed by the fusion of egg and sperm. So zygote and any other cell after this process, okay, now it has 46 chromosomes. After the fertilization process is over, okay, after the fusion, the zygote, it contains 23 pairs of chromosomes, and totally we have 46 chromosomes. And after it, the zygote, it will replicate like normal cells. Okay, it will keep on dividing, and a new individual is formed through this replication. Is it clear? Now there is something differential gene expression. See, everything is forming, a complete individual is forming from the zygote. Okay, zygote is dividing into many cells, and from those cells the complete human body is formed. Different organs, okay, having different functions, they are formed in this process. So when all the cells of the body, they contain the same 23 pair of chromosomes, okay, why do they behave differently? So a given cell type, in a given cell, okay, only a particular set of genes, they will express themselves. Okay, in given cell type, only a particular set of genes, they will express themselves, and the functionality of those organs, it will be different. So precisely for this reason, we have a variety of organs inside the human body, and this is in the crudest form, we can say that it is the differential gene expression. Okay, so this is differential gene expression. And what is a gene? What is a gene? Then what is genetic material? DNA is the genetic material. Okay. See, I told you about the nucleotide. I told you about the nucleic acid. I told you about the nucleotide. So these genes are basically the functional sequence of the nucleotides. The functional sequence. Functional sequence of nucleotide is a gene. Okay, If you will change the arrangement of the nucleotides in a chain, then it will not be the same gene anymore. A functional sequence, okay, a functional sequence of the nucleotide is called as a gene. Yes, plus gene is a unit of inheritance, and I told you about the DNA, and DNA was a nucleic acid, and it is formed of the chain of nucleotides, and the functional sequence, the functional sequence of this nucleotide that is serving a special purpose, we call it a gene. Yes, basically genes, they are part of the DNA. Yes, Palak, because they are formed of nucleotide, and nucleotide is the it is forming the whole DNA, and gene is a functional sequence on the nucleotide. Yes, Palak, gene is itself gene is a segment of DNA, and that too it is a functional segment. Not any segment of the DNA is a gene, but it is a functional segment. And if there is any sudden change in the sequence of the genes, then that is known as mutation. A sudden change in the sequence of the genes is known as mutation. Okay. Ritika is asking, why isn't it a part of RNA? See, I think RNA is a genetic material, but RNA is not part of our cell. I don't think so. It is part of the human cells. Yes, RNA is also genetic material, but it is not found inside the human cells. Differential gene expression clear hai? Okay. All the body cells, they have the 23 chromosome, same pair of 23 chromosome, okay, which are identical, but we have different organs performing a different function. So only some of the genes, they express themselves in a particular set of organs, in particular set of tissues. Okay, so the same type of cells, they are working differently. This is the differential gene expression, and we have some 30,000 plus genes that have been identified. Some 30,000 plus genes, they have been identified inside the cells of the human body. Okay. Yes, Meenu. Mutation is related to the genes. Okay. If the sequence of the nucleotide it is changing, gene is basically a sequence on the nucleotide, a functional sequence on that chain nucleotide. So if there is some change in that functional sequence, then we'll say that the gene, there is some tampering, there is some alteration with the gene. And that alteration, sudden changes in the genes, is known as mutation. So in simple words, mutation is a sudden change in the genes. Sequence जैसे उलट पुलट हो जाएगी, जीन की फंक्शनैलिटी ही चेंज हो जाएगी. It will serve a different purpose. This human genome project, it is related to the collection of the genetic material of the humans. It is related to it. So a sudden change in the genes is known as mutation. Now the concept of twins. Okay, these twins, I explained you the birth process. Okay, the one sperm, it fuses with one egg. One sperm fuses with one egg, and in this process, okay, the zygote is formed. Zygote is formed by the fusion of egg and the sperm. Yes, Nathan is absolutely right, Nikhil. The genes, they can be suddenly changed, okay, by interacting with the electromagnetic radiation, okay, by the change of environment, by heat, temperature, or anything. And they define it as the sudden change. Mutation, it is a random process. Okay. Yes, Ashwarya. The twins, they are of two kinds. We have monozygotic and we have the dizygotic twins. Okay. Yes, Nikhil. UV radiation, it can also alter the genes. See, a gene, basically, these are all the molecules, biological molecules, organic molecules. And you know that electromagnetic radiation, the UV rays, it can break the bonds between these molecules. So UV radiation, they can alter the gene sequence. Okay, it can cause the mutation in the genes. So welcome, Nikhil. Now, normally what happens, a female releases only one egg per month. Okay, only one egg is released per month. But in case of males, there are around 500 million sperms. Okay, that are produced. So, in the formation of zygote, only one of the sperm, it fertilizes the egg. So we have millions of sperms, and out of these millions of sperms, the one will be able to fertilize the egg. Okay, and the fertilized egg, it is known as zygote. And zygote, it leads to the birth of a single baby. Okay, this is the normal process. Then, why do we observe these twins? Okay, there is some reason for it. If a female by chance produces two eggs, okay, already there are millions of sperms, so both the eggs, they will be fertilized, and in that case the two zygotes, they will be formed, okay, and two babies, they will be born through this process. So these babies, they will not be identical because two separate zygotes were formed. And the Aishwarya here, she has already termed it as the dizygotic. So such twins, they are known as dizygotic. Okay, because these twins, they are a product of two zygotes. These are the dizygotic twins. ठीक है, एक egg produce ना होके दो egg produce हो जाएंगे. And because of that aberration, the twins will be born. So these dizygotic twins, they are non-identical twins. Okay, वो एक दूसरे से एकदम हूबहू एक दूसरे जैसे नहीं होंगे. They will be just like the normal siblings. Like we have normal siblings. So there will be similarities between them. Okay, and those similarities will be similar to the normal siblings. So non-identical twins and siblings में कोई खास difference नहीं है. And these dizygotic twins, they are also known as fraternal twins. Okay. So why the two eggs they are producing? What is the reason that the two eggs are produced? See, there are two hormones in the females. One is the estrogen and one is the progesterone. Okay, so these are the two hormones, and estrogen is estrogen and progesterone progesterone. So these are two hormones, and these hormones inhibit the production of second egg. But if there is something wrong with the production of these hormones, then the second egg it can be produced. Okay, some mechanism was preventing the production of the second egg, and that mechanism was these hormones. We have the estrogen and we have the progesterone, and if there is something wrong with the production of these hormones, a second egg it can be produced, and we can have the non-identical twins or the dizygotic twins or the fraternal twins. Okay, clear? See, the non-identical twins, they are born because two zygotes are formed because of the production of two eggs. Usually the females they produce only one egg, but because of the there some abnormality with the production of estrogen and progesterone, the hormones that were responsible for controlling the production of egg, the two eggs were produced. Okay. क्योंकि एस्ट्रोजन एंड प्रोजेस्टेरोन ने ठीक से वर्क नहीं किया, ओके, दे वर नॉट प्रोड्यूस्ड इन द एम्पल क्वांटिटी, सो उस वजह से दो एग्स प्रोड्यूस हुए, एंड दो एग्स प्रोड्यूस हुए, इसीलिए वी हैव द टू zygotes एंड वी हैव टू बेबीज. Okay, now we have the other type of twins. We have the identical twins. The identical twins will be like these boys. They are completely identical to one another. Yes, Fareed. See, the identical twins, they are formed by one egg plus one sperm. Okay, the one egg plus one sperm, it will produce a zygote. Okay, and by some process, okay, if this zygote it bifurcates into the two similar zygotes, then we will have two similar kind of zygotes. By the fusion of one egg and one sperm, a zygote is formed, and that zygote it gets divided into two similar zygotes. Okay, so two new individuals, they will be developed from these two zygotes. Okay, then in this case, because two individuals, they are originating from a single zygote, they are known as the monozygotic twins. ठीक है, एक एग और एक स्पर्म मिलके एक zygote बनाएगा, and by some process that zygote it will split up into two parts, two identical parts, and from those two identical parts, two new individuals they will be born. So this process we have in this process, because two individuals they are born out of the same zygote, so this is the monozygotic twins, and these twins they will be identical to each other. Okay, barring the fingerprint, except the fingerprint, so all of their traits they will be same. Even 100% of the DNA it will be same. Okay, the 100% of the DNA will be same. So identical twins, they are they are exactly identical to each other, and twins that are born from two separate zygotes, they will not be identical. Only the twins that are born out of a single zygote, they will be identical. Kritika, fingerprints, okay, fingerprints, they are not related to the genetic material. Fingerprints, they are different. I do not, okay, I do not know why, but fingerprints, they do not relate to the genetic material. The maximum part of the DNA, it will be the same. Okay, there will be some minor changes because of the external factor, but because the same single cell, it is replicating twice. Okay, the same genetic material, it is inherited by the two cells. So at that level, the DNA is the 100% same. If at the level of the cell division, there is something different. Why does the zygote gets divided? That also I do not know, and I think even the even the doctors cannot explain it satisfactorily. Because of some reason, some instability, okay, some minor changes in the environment, okay, it can happen that a zygote gets divided into two parts. So the identical twins, they are the monozygotic twins, and if a zygote is breaking into two parts, then we will have the identical twins. But if the zygote it breaks into three parts, then the birth it will be abnormal. Okay, the birth will be abnormal if the zygote splits into three parts and three identical parts. And identical twins, they have to be of the same gender. Okay, because if this contains the X chromosome, then it has split into two parts. These two cells, they are the identical copies of this cell. Okay, in this replication, these two cells, they are the identical copies of this cell, the parent cell, and for this reason, the identical twins, they will be of same gender. And the non-identical twins, they are born out from the separate zygotes. So in their case, their gender can be different. Are you getting my voice? You people are typing something for quite a long time. Okay, Palak has explained the reason for the fingerprints. Okay, Palak, because that is a random process, so the fingerprints, they will be different every time. Okay, so that is the primary reason. Palak has given one answer, that as the fetus grows inside the womb, and okay, because of some kind of frictional events inside the womb, the fingerprints, they are a result of those processes. Okay, Harini, thanks for that information. But at the crudest level, if the zygote it gets split into two parts, okay, two identical parts, It is similar to the fusion reaction. So one thing, it is getting split into two parts, two identical parts, and from those two identical parts, okay, two new individuals, they are growing from those parts. So even the identical twins, they have the different fingerprints, and because they are derived from a single cell, okay, which bifurcated into two parts, two identical parts. So for that reason, okay, we say that even their genetic material, the DNA, it will be 100% same. And because of the surrounding factors or any other factors, okay, there may be minor variations to it, but technically they should have the same genetic material. Because it does not matter if you are taking samples from my skin, if from this side of the face you have taken one sample and from this side of this face you have taken one sample, then the genetic material will be same from these processes, because as Aishwarya has pointed out, they are formed by the reaction of from the division process, which is known as mitosis. So let us discuss the twins. Even the clones, okay, even the clones, they have the different fingerprints. Clones are basically the the carbon copy of an individual. Clones are the carbon copy of an individual, and even the clones, they have the different fingerprints. So the DNA matching, the DNA of an individual, it should match 50% to their parents, but in case of identical twins, the DNA, it will match the 100%. Okay, and even in case of the clones, the DNA, it will match 100%. And the DNA of any individual, it will match to his parent by a factor of 50%. Okay, now we have twins which are named as the conjoined twins or the Siamese twins. So these are the twins who are born attached. Okay, their bodies are attached to each other, and they are known as the conjoined or Siamese twins. And this is due to the incomplete splitting of the zygote. If the zygote, okay, it is not splitted properly, then from the incomplete splitting of the zygote, the two individuals are born, okay, and the certain body parts, they are joined to one another. So Siamese twins, they are a result of partially divided zygote. Okay, and that partially divided zygote, it gets converted into two babies, and certain body parts, they remain attached to one another. As you can see in this picture, these two twins, they are attached to one another because of the incomplete splitting of the zygote. Now Aishwarya has pointed out two kinds of processes. One is known as the mitosis and one is known as the meiosis. Okay, so in the mitosis, a single cell, it splits into two similar cells. These are the identical copies. Okay, one cell, it divides into two cells. Basically, mitosis is a cell division process, and meiosis is also a cell division process. So here the identical copies are produced, and again this cell will split into the two identical copies. So this is the mitosis. This process is mitosis. So mitosis is equational cell division. Okay, and the number of chromosomes before and after this process is the same, because the same copies are produced. So in the copy also, the chromosomes, they will be same. Yes, Aishwarya. See, if there is a cut on my skin, then the skin will repair itself after some time, and in that reaction, the skin will repair itself through mitosis. Okay, so in the normal body cells, they are known as the somatic cells. Soma means body, S-O-M-A, soma means body, and the somatic cells, it means the body cells. Okay, so in the normal body cells, they grow by the process of mitosis. कोई भी चोट लग जाती है, then the skin it reappears and the organ they repair themselves by this mitosis cell division. The cell they grow by the cell they divide and the process is the mitosis. And the same type of cells they are formed in this process, and each cell contains the same number of chromosomes. The chromosomes they are same before and after the process. Okay, this is simply the carbon copy. But in case of the meiosis, it occurs in case of the spatial cells, which are the eggs and sperms. So meiosis occurs only during the formation of eggs and sperms. And meiosis के अंदर हमने बात की थी that in a cell, in every cell, we have the 46 chromosomes. But after the meiosis, when the egg and sperm is produced, the egg or sperm they contain only 23 chromosomes. Okay, only half the number of chromosomes is present in the egg or the sperm. And this type of cell division is known as meiosis, and meiosis occurs in the sex cells. Okay, mitosis occurs in the normal body cells, and meiosis occurs in the sex cells. And meiosis is a reductional cell division because the number of chromosome reduces. Okay, from the 46 chromosomes in the sperms and in the egg, we have only 23 chromosomes. So the number of chromosomes is getting reduced to half due to the cell division. Mitosis, meiosis, clear? Nitin, what is the full form of this SS? You are writing SS. What is it? Okay, Nitin. If it means yes, then it is fine. Then we have some kind of chromosomal aberrations. Okay. If there is any error in the number of the chromosomes or in the structure of the chromosomes, then that thing is known as chromosomal aberration. ठीक है, क्रोमोसोम की स्ट्रक्चर में या फिर क्रोमोसोम्स के नंबर में कोई भी चेंज हो जाएगा, that is the chromosomal aberration. If there is any change in the number of the chromosomes or in the structure of the chromosomes, that is chromosomal aberration. And because of this chromosomal aberration, we can have various kind of diseases in the humans. Yes, we can call it, okay, it can be due to some sort of mutation. Yes, Ashwarya. So one is the super female syndrome. Okay, in case of super female syndrome, we have the 47 number of chromosomes. The number of chromosomes is 47, and in case of females, we know that we have 2x chromosomes. Okay, but if instead of having 2x chromosomes, we are having 3x chromosomes, then this super female syndrome it will develop. Okay, we are having the 47 chromosomes and we are having one extra X chromosome in case of a female. Okay, if any female has one extra X chromosome, then that female is suffering from the super female syndrome, and such women, they are sterile. Okay, so its symptom is that these such persons, they are sterile, and it is known as super female syndrome. And likewise, we have the super male syndrome if there is one extra Y chromosome. Okay, X and Y is the male, and if we have one more Y chromosome, then we have the super male syndrome. And these individuals, they have high criminal attitude, and this is one of the major symptoms. they have the criminal attitude, a tendency to commit crimes frequently. So those juveniles, the juvenile delinquent, it is usually associated with those individuals, those young people, okay, who frequently commit the crimes. So the juveniles who frequently commit the crimes, it is found that most of them, they are suffering from the super male syndrome. Okay, super female syndrome के अंदर there is one extra X chromosome, and in super male syndrome we have one extra Y chromosome. In दोनों cases में number of chromosome is 47. Okay, we have 47 chromosomes. In both of these cases, in super female there is one extra X chromosome, and in the males we have one extra Y chromosome. Then we have this Turner syndrome. In case of Turner syndrome, we have only 46 chromosomes. We have 45 chromosomes. These chromosomes ultimately they define the behavior of an individual. Okay, because the chromosomes they contain the genetic material, and your genes, they are basically like the programming inside you. Okay, so having one extra X chromosome or having one extra Y chromosome, it can alter your behavior, and it is found that the juveniles who are engaged in crime, who are habitual offenders, okay, they are mostly suffering from this super male syndrome. They are having one extra Y chromosome. Okay, there is something wrong with their programming, their biological programming. It is different. Got it, Nikhil? Okay, the set of instructions that is guiding your behavior, it is stored, okay, inside your genes. That I do not know, Nikhil. इसके लिए ऐसे किसी पर्सन से मिलना पड़ेगा तुम्हें. Now in case of Turner syndrome, we have only 45 chromosomes. Okay, we have one X chromosome, and there is a vacancy for the 46th chromosome. So one chromosome is missing. One chromosome is missing in case of Turner syndrome. Okay, one sex chromosome is missing, and such people, they are mentally retarded, and because one X is present, okay, so these are also sterile female. They are mentally retarded. Okay, Palak. Nikhil, Palak is saying that such individual juvenile delinquent person, they are okay, they are very normal in their sexual activity, fertility, but they are more aggressive because of one extra Y chromosome. Okay, they are more aggressive, and for that reason they commit crimes frequently. See, mentally retarded person cannot commit crime. Okay, to commit crime, there must be some level of smartness in any person. Now there is one thing which is known as Klinefelter syndrome. In case of Klinefelter syndrome, we have two X chromosomes, and there is one extra Y. So we have the XXY, and because of one extra X, okay, X and Y is male, and because of one extra X chromosome, okay, these males they have certain female characters. Okay, because of one extra X chromosome, these males have certain female characteristics, and these males, they are also sterile. Okay, so Klinefelter syndrome, it is a male which is X and Y, and this male has one extra chromosome which is X, and because of the extra X chromosome, okay, this male develops certain kind of female characters, and this male is sterile. Now we do not have any Y syndrome, Y and 0, Y naught syndrome. We do not have any Y naught syndrome, and the reason for this is that we cannot survive without the X chromosome. Okay, and if you do not have the X chromosome, you will die a zygotic death. The individual will die as a zygote only. If the zygote does not have the X chromosome, the zygote will die. Okay. Without the X chromosome, the zygote, it cannot develop into a full-fledged individual. Now, in case of transgenders, either there is some abnormality in the sex chromosomes, okay, some kind of changes in the sex chromosomes, or there will be some changes in the sex hormones. Okay, so the problem, it can be related to either of these. Either there is, yes, Nikhil?
The transgenders, they are real, sir. Actually, they exist really, or that is just fake. That is simply.
Nikhil, transgenders, they are real, bro. Okay, Nikhil, transgenders, they are real. Even we have protected the transgenders' right. We have passed some bills in the parliament for the protection of the transgenders' right. Okay. Now we are saying that because of Turner syndrome, identical twins with different gender are born, very rare case. But the people with the Turner syndrome, they are sterile. Okay. The people with the Turner syndrome, they are sterile female. Now we have this Down syndrome. Okay, and Down syndrome is because the 21st chromosome, it is present in 3 copies. If the 21st chromosome, we have 3 copies of the 21st chromosome, then we will have this, the individual will develop the Down syndrome. ठीक है, 21st chromosome की 3 कॉपी हो गई. Usually we have the pair of the chromosomes, लेकिन अगर उसकी 3 कॉपी होगी, then we will have the Down syndrome. And because of the Down syndrome, its symptoms are mental retardedness. Okay, your IQ level, it will reduce drastically. It will be below 50, and the individual will be infertile. Trisomy 21. It is the autism. Okay, and this is also known as the Mongolian idiocy. And because the facial features of the individual, they resemble the Mongoloids. They resemble the Mongolians, and that is why it is also known as Mongolian idiocy. Okay, and its symptoms are the symptoms of Down syndrome, the IQ level below 50, and retardedness, and the infertility. And there is also one more syndrome, which is the cat's cry. Cat's cry is cry do chat, and in this cat's cry there is some alteration, some aberration with the 5th chromosome, and in fact the 5th chromosome is missing, and again its symptom is mental retardedness. In case of Down syndrome, okay, there is one extra copy of the 21st chromosome, so we have the 47 chromosomes. So one thing we have realized that if there is something, okay, some aberrations inside the chromosomes, then the functionality inside an individual, it will change drastically. It can even give birth to the retardedness, and it can make a person infertile. Okay, so the chromosomes, they are the genetic material, and they contain a lot of information about the individual. Now let us talk about the cloning. Critical case of transgenders, we are saying that either there is some abnormality with the production of the hormones, or there is some abnormality with the chromosomes. Okay, we cannot say certainly that they have 46 chromosomes. Either the hormonal issues are there, or some kind of aberrations in the chromosomes are there. If there would have been any specific term, then we would have covered it, but there is no specific term for the, okay, in case of the transgenders. But transgenders, they are usually associated with some kind of abnormality in the hormones as well as in the sex chromosomes. Now let us move on to the cloning. See, cloning is basically what is cloning? What is your perception of cloning?
How that?
Sir, cloning means, according to me, it's like crossbreeding, sir.
Cloning is crossbreeding. No, like cloning is not crossbreeding.
Like taking one's genes and the other person.
Nikhil, it is like the photocopying. Okay, it is about making the identical copies of any organism.
So cloning is the process in which we produce morphologically and genetically identical individuals.
Yes, Palak, very good. Morphologically and as well as genetically. She has used the very apt terminology, morphologically and genetically. So morphology means in the physical appearance, okay, the organism will be identical to the parent organism, and also the genetically, the genetic material, it will also be same. So how it is not an artificial process of copying of genes. Basically, it is the manipulation of the birth process. Okay, we are manipulating our knowledge of this whole process. Okay, so how we manipulate this, I will explain you. See, in the normal birth process, what happens? We have the this is the egg cell, female egg cell. And in the female egg cell, we have only 23. Okay, only 23 chromosomes are there in the female egg cell, and we have the sperm, the male cell, and in this we have the 23 chromosomes. So when they fuse, what happens is, and obviously in this cell we have this cytoplasm. So this cytoplasm is the cytoplasm of the egg cell. the cytoplasm of egg cell. Then in the fusion process, what happens? The nucleus, it enters the female egg cell. Okay, and rest of the cell is discarded. Only the nucleus with the genetic material 23 chromosome, it enters the female cell, female egg cell. Okay, and now what do we have? We have the 23 chromosomes of the female, and we have the 23 chromosomes of the male, and this is having the cytoplasm of the egg. Now in this zygote, in this fusion product, we do not have the cytoplasm of the male. Everyone is with me? We do not have the cytoplasm of the sperm cell. We have the this contains three things. This contains the genetic material of the egg from the egg cell. It contains the cytoplasm from the egg cell, and it contains the genetic material from the male cell, from the sperm. Okay? सबको समझ में आया? Ritika, I am saying when the sperm cell fuses with the egg cell, egg of the female, this is the egg cell, and this egg cell contains 23 chromosomes. Okay, and this is the cytoplasm inside the egg cell, the female egg, and we have the sperm, we have the nucleus containing 23 chromosomes. Okay, and there is the cytoplasm of the male cell. So when the fusion occurs, only the genetic material, okay, it enters the female egg cell, and rest of the cell is discarded. ठीक है, इस सेल का, स्पर्म सेल का साइटोप्लाज्म और बाकी जितना भी सेलुलर पोर्शन है, it will not enter the female egg cell. Only the 23 chromosomes, the nuclear part, it will enter the female egg cell. Okay, so finally the zygote that is formed, the zygote contains the cytoplasm, and the cytoplasm is of the egg. This is the cytoplasm of the egg. Zygote के अंदर cytoplasm किसका है? Egg cell का. And it has the 23 chromosomes from the female, and it has 23 chromosomes from the male. So zygote has the cytoplasm, female का cytoplasm, female genetic material, and the male genetic material. Okay, clear? Got it. So this zygote, it is consisting 46. It is made up of 46 chromosomes. There are 46 chromosomes inside the zygote. Very well. We have 46 chromosomes, plus we have the cytoplasm of the female egg. We have the cytoplasm of the egg, plus we have the 46 chromosomes. The complete set of 46 chromosomes are there. Now in cloning, we manipulate this process. In cloning, what we do? I talked about the somatic cells, the normal body cells. Okay, we will take the normal body cell of an individual. Suppose we have person A, and we have taken the normal body cell of the person A, and in person A का हमको clone बनाना. Okay, we have to make a clone of the person A, and suppose this is the egg cell with 23 chromosome of any female, and this female is a donor. She is a donor for the cloning process. So now we do not have the sperm cell, we have a donor egg cell. Okay, and again it contains the cytoplasm, and it contains the genetic material. 23 chromosomes are there, and we have to make the clone of the person A. We have taken a normal cell, okay, a normal cell of the person A, normal body cell of the person A, and we know that normal body cell, they contain the 46 chromosomes. ठीक है ना? सबको पता है that normal body cells they contain the 46 chromosomes and we have the cytoplasm. Okay, obviously any cell has cytoplasm. Now in the cloning process, what we will do, we will remove the nucleus of this cell. Okay, we have removed the nucleus, we have this nucleus, and we will remove the nucleus of the female cell also. These 23 okay chromosomes, they will be discarded, they will be removed from the cell and they will be discarded. Now this donor egg cell, it only has the cytoplasm of the egg cell. Okay, now we are putting this genetic material consisting of 46 chromosome. Zygote should have 46 chromosome. We are putting this number of chromosome inside the egg cell. Egg cell का nucleus तो निकाल के फेंक दिया, and we are putting inside the nucleus containing the 46 chromosomes. Okay, and this nucleus is, it belongs to the person A. It has the genetic material of the person A. Okay, so this is a zygote. This is a zygote and it contains the genetic material of the person A. And we will implant this zygote inside any female body and it will develop into a new individual which will be similar morphologically to A and it will be similar genetically to A. Okay, समझ में आया? Very good. So in case of cloning, what we are doing is we are discarding the genetic material of the egg cell, and inside the egg cell we are putting the 46 chromosomes जिस बंदे का हमको clone बनाना है. The person whose clone we have to make, we will take his genetic material, we will take his 46 chromosomes from any healthy body cell, okay, and we will put those genetic material inside the egg cell. Okay, and after that, that zygote will be implanted inside any female body and it will develop into a new individual. Okay, and that individual will be similar to the person A. Kritika, any somatic cell, we need any somatic cell, any part, any body cell can be used for this purpose. We just need the genetic material. Meenu Singh, sex chromosome will be there. So we are taking the 46. Take any cell of my body and any cell of my body contains the 46 chromosomes. Okay, and in those 46 chromosomes, one of the chromosome will be X and one of the chromosome will be Y. Okay. We do not need a sperm. There are some ethical issues associated with cloning. Okay, that is why the cloning is banned. So let us look at it one more time. So what the donor has to be, okay, it can be any female. We need an egg. The cytoplasm of the sperm cell is of no use. Okay, it is of no use in cloning. We need the cytoplasm of the egg cell. So cloning is the literal meaning of cloning is copying. Okay, and the zygote, I have already explained that the zygote contains the cytoplasm of the egg cell and it contains the nucleus from the egg and it has only the nucleus of the sperm. Okay, the cytoplasm of the sperm is discarded. Very well. Then in the cloning process, we will take the egg from any donor and obviously the donor will be a female and we will remove the nucleus from the donor egg cell. Okay, we will remove the nucleus from the donor egg cell and in the vacant egg cell we will put the nucleus from a somatic cell of the person. Somatic cell, they are the normal body cell. All these cell, okay, except the sperms, they are the somatic cells inside the body. So we will put the nucleus from a somatic cell inside the vacant egg cell. This is point number 2. Then the resulting zygote is the cloned zygote. Okay. Then this resulting cloned zygote, this resulting zygote, it will be put inside the body of any female person, and that person is known as the surrogate mother. Okay, we will implant this zygote inside a female body, and the resulting baby will be the clone of that person. So this is the whole process of cloning. And the first cloned organism was this sheep, and the name of this sheep is Dolly. Okay, and the first clone of a cat is named as the copycat. Okay, and in 2002 there are reports that a Bahamas-based company, and name of this company is Clonade, they created the first human clone, and the name of that clone was Eve. Okay, and although the human cloning, it is not permitted legally, it is banned, and there are certain issues with it, that it might impact the population, and it might be used for the criminal purposes, and the clones, they have a shorter lifespan. The clones, they have a shorter lifespan because the somatic cells are used. Okay. So do you have any questions? This is all from my side. For today, this is all, and tomorrow we will continue with the biotechnology. The clone of a cat, Billy ka clone. See, cloning is genetic engineering. Means cloning, we cannot say it is genetic engineering because we have not modified the genes. It is not genetic engineering, but definitely it is biotechnology. Genetic engineering involves some kind of manipulation with the genes. We have not manipulated the genetic material. Okay, something known as the designer babies. Okay, by modifying the genes, we can get the desired traits inside the babies. So those designer babies, they are a part of genetic engineering. And the BT brinjal, BT cotton, they are a part of genetic engineering. But I think cloning is not part of genetic engineering because we have not altered the genetic material. We have only manipulated the birth process, the normal birth process. We have manipulated it, and we observed that the zygote contains the 46 chromosomes, and 23 of them belong to the mother, and 23 of them belong to the father. So what they have done is they have discarded that genetic material, and they have put the genetic material 46 chromosomes of the person whose clone we have to make. No, cloning is not allowed. Okay, cloning is not allowed. It can be allowed in the countries such as North Korea. Okay. In the democracies of the world, the cloning, it is not allowed. It is considered as an unethical practice. Okay, so this is all for today, and we'll meet tomorrow at 3 p.m. And tomorrow, I think 2.5 hour session I will take for the biotechnology, and the rest of the time I will take up your doubts regarding the blockchain. Okay. So this is all for today. Thank you from my side and see you tomorrow. Goodbye.